Hello and welcome. There's this major tension in the world of scientific research. On one hand, you've got the promise of these incredibly powerful, easy-to-use software tools. But on the other, you have the absolute non-negotiable need for scientific rigor. So the question is, how do you get one without sacrificing the other?
And this is really the central question for so many scientists, PIs, and lab managers, isn't it? There's this kind of nagging fear that no code is just another way of saying black box. That if you can't see the code, you just can't trust the results. And that is a totally valid concern, and it's one we're going to tackle head on.
So, to see how a platform like STARS deals with these issues, we're going to break this down into 4 key areas. First up, building trust through transparency. Then, how it can supercharge your team's workflow. Third, connecting different kinds of data and, just as importantly, people. And finally, how it smooths out that whole path from raw data to a finished publication.
Okay, let's start with the biggest hurdle of all, trust. We're going straight at that black box problem, that fear that a tool's simplicity is just hiding a lack of scientific depth.
Now, what's so interesting here is you can really see the 2 different philosophies laid out. A lot of no-code tools, well, they rely on proprietary, opaque methods. You put your data in, you get a chart out, but how it got there is a complete mystery. STARS takes the exact opposite approach. Every single analysis is built on standardized, peer-reviewed statistical libraries, the ones the scientific community already knows and trusts. There are no secret formulas here.
And this really gets to the core of that transparency, reproducibility. For every version of the software, there's comprehensive methods documentation. And here's the crucial part. You're not just getting a result. You are getting a fully citable methodology that meets tough standards like GXP. That ensures your work can actually be replicated and trusted by your peers.
Okay, so with that foundation of trust in place, let's look at how this can actually supercharge your research team. And listen, this isn't about replacing your experts. It's about solving that team bottleneck that we've all experienced.
This new workflow is, honestly, a game changer. So instead of every little request going to your bioinformatics core, now the wet lab scientists can do their own initial data triage. The QA, QC, some basic clustering. This frees up your bioinformatician to focus on what they do best, building those really complex, high-level custom models. And for the senior PI? Well, it means they can test ideas the very moment they have.
This quote just captures the power of that so perfectly. I mean, think about it. Inspiration doesn't keep a 9-to-5 schedule, right? The ability to test a hypothesis right away, to immediately explore a new idea without creating a new task for somebody else, that fundamentally changes the whole pace of discovery.
And that new pace of discovery leads us right into our third section, connecting data and connecting people. Modern science is all about collaboration, but that collaboration can get stalled by data silos and what we call the jargon gap.
Oh man, this line perfectly illustrates that jargon gap. We've all been there. Everyone is absolutely brilliant in their own field, but they're all speaking slightly different languages. The exact same word can mean totally different things to different experts, and that leads to miscommunications that can slow down an entire project.
So how do you bridge these gaps? Well, first you have to bridge the data silos. STARS is built for something called multi-modal fusion. What that really means is you can take quantitative data from, say, an imaging platform like brain metrics from Qmenta and merge it directly with flat data like protein expression levels or clinical scores. It lets you ask questions across all your different data types.
And here's the solution to the people problem, a visual-first interface. Instead of everyone arguing over terminology, the entire team, the biologist, the statistician, the clinician, is looking at the same interactive volcano plot or heat map. The visualization itself becomes the shared language. It allows for a conversation that's centered on the data, not the jargon.
Okay, so we've established trust and we've improved teamwork. For our final section, let's get really practical and look at the whole research lifecycle, from the moment you get your data in all the way to getting those publication-ready results out.
So the whole process has to start with a frictionless data import. The platform accepts the standard formats you're already using, CSV, Excel, text files. But crucially, it's also optimized for those really complex data structures we see in modern research, like the wide and long data you get from bulk RNA-seq, proteomics, or longitudinal clinical trials.
And then at the other end of the pipeline, you get publication-ready exports. And this is a huge time saver. Every chart, every plot can be exported in high-resolution vector formats like SVG or PDF. You can customize the colors, the labels, the scales right there in the interface to meet the specific requirements of top-tier journals like Nature or The Lancet. No need for a separate graphic design tool, it's all right there.
So we'll just end with this thought. When you remove all those technical and collaborative barriers, the fundamental question really changes. It's no longer about if you can test your idea. It's about how quickly you can go from a simple question to a real discovery.